Hello, everyone, and welcome to today's webinar. My name is Olga Vodi, and I'm a SYNGAP parent and part of the team at SYNGAP Research Fund. Our presentation today is evaluating a stem cell gene therapy approach for SYNGAP1. And I have the pleasure to introduce today's speaker, Dr. Joe Anderson at the University of California, Davis. Dr. Anderson is the Associate Professor of Internal Medicine in the Division of Infectious Diseases at UC Davis. He has been in the field of stem cell gene therapy for the past 20 years and has developed numerous therapeutic lentiviral vectors for HIV, Tay-Sachs-Sandoff disease, and Angelman syndrome. He's evaluated their safety and efficacy both in vitro and in vivo in adult and pluripotent stem cells. He performed the in vivo safety efficacy data for the current pre-selective anti-HIV lentiviral vector being used in a phase 1 clinical trial for HIV lymphoma patients. Excuse me. Dr. Anderson has also developed stem cell gene therapies for both Tay-Sachs-Sandoff and Angelman syndrome that are currently in the investigational new drug enabling experimental stage. Dr. Anderson has successfully written, submitted, and received approval of NIH RAC, pre-investigational new drug, and investigational new drug applications for his HIV stem cell gene therapy work. A recorded version of this webinar will be available on the SRF website under webinars in the family menu. By the end of this presentation, you will have the opportunity to get to answer your questions. We'd love to hear from you, so please write those questions in the Q&A below. For those of you just joining us, welcome. And again, our speaker is Dr. Joe Anderson, and his presentation is on evaluating stem cell gene therapy approach for SYNGAP1. It's now my pleasure to turn things over to Dr. Anderson. Thank you.
Thank you, Olga, and thank you everyone from SRF for inviting me to give this webinar on our research. So, as Olga mentioned, my name is Joe Anderson. I am an associate professor at the University of California, Davis. My research focuses on stem cell gene therapy for multiple diseases, from infectious diseases to lysosomal storage diseases. And now we've moved those strategies and approaches into some neurodevelopmental disorders to evaluate whether this approach will work for those disorders as well. So today I'm going to talk to you about our stem cell gene therapy approach as a potential treatment for SYNGAP. The way that this approach would work is through a mechanism called cross-correction. So instead of directly gene modifying neurons, we would actually be using microglia cells that are in the central nervous system to transfer wild type and functional SYNGAP to the affected neurons. So this has been successfully used for some lysosomal storage diseases and leukodystrophies that are currently in clinical trials. We've shown that this works well for Tay-Sachs disease, which is a lysosomal storage disease, and then also for Angelman syndrome, which is a neurodevelopmental disorder. The way that this would work is the microglia cell, which is a progeny from the hematopoietic stem cell, which is the blood-forming stem cell. That would be the cell that would be genetically modified with the functional and wild-type form of SYNGAP. These cells home into the central nervous system where they're used as accessory cells, and they'll be able to express SYNGAP and then transfer it to affected neurons. So that's the overall strategy scientifically and in our hypothesis on how this would work. So we wouldn't be directly reprogramming or genetically modifying the neurons, but we'd be using these microglia cells as protein factories to then transfer the functional protein to the neurons. The type of gene therapy that we use is called insertional gene therapy. So we're actually permanently integrating the wild type and functional copy of SYNGAP into these blood stem cells. So it will be in there for the life of the patient, and every cell that's derived from that stem cell will have that copy of that gene as well. And so we'll insert this copy of SYNGAP using a lentiviral vector. So this vector system has been engineered to allow permanent integration. It's derived from the HIV system, where HIV permanently integrates into the genome. However, these lentiviral vectors are stripped of all the pathogenic genes that HIV has. The modifications made to the vector system also don't allow the vector to replicate once in the cell. So they're called self-inactivating vectors. And so they are still able to integrate their genetic information into the genome of the target cell, but they're not able to replicate. So it's more like a one-way street. We're able to get the SYNGAP gene in with its respective promoter to drive expression, but then that's all the vector will do. It will drive expression of the SYNGAP gene. The cells will make the protein, and that's all that the vector will do. It won't be able to replicate. It doesn't have any of the components that could cause HIV. This vector system has been used in numerous clinical trials for other diseases. So it's a safe method of inserting a gene that you want into a target cell. The reason why we wanted to use the hematopoietic stem cell is that the blood system and the blood system cells are found throughout the body, even in the CNS. So as I mentioned, the main cells that will be doing the work for this approach are the microglia, which are found in our brains. They are accessory cells, just like the other glial cells. And so they know to home into the brain and then do the work that they will do. And then in what we want to do for SYNGAP is to program them to express the functional copy of SYNGAP, transfer it to the neurons, and then the neurons will be able to use this protein for normal function and hopefully correct clinical phenotypes. This is a classic tree of hematopoiesis. It shows the pluripotent stem cell at the very top, and this is the hematopoietic stem cell. This stem cell resides in our bone marrow. There are a very small percentage of the cells that circulate throughout the blood, but then eventually they go back into the bone marrow. That's their home. That's where they reside, and they constantly make our red blood cells and our white blood cells, which are immune system cells, for the life of us. Babies, 80-year-olds, all of us have these cells in our body because we constantly have cell turnover. And so our immune system has to constantly make new immune system cells, including red blood cells. And so if we're able to target this hematopoietic stem cell with our gene therapy, there's the potential of it being just a one-time treatment. If we're able to get enough of these hematopoietic stem cells gene modified with our vector and our SYNGAP protein, then the body would make all of these progeny and daughter cells that express the SYNGAP protein for the life of the patient, including the microglia in the CNS that will transfer it to the neurons. And that's the reason for targeting the stem cell. We could target these other daughter cells and the microglia themselves, possibly transplant them into the brain, but those cells do have a half-life, they do turn over, and so we would have to repeat that procedure potentially a number of times. But if we target the stem cell, then potentially there's just a one-time treatment, because those cells constantly make our immune system cells, but also make more of themselves. So there could be an arrow up top, at the very top by the stem cell, kind of pointing back at itself, because it's not only makes more of these immune system cells, but it also makes more of itself so that you have them for the life of the individual. So this is just a basic schematic of what we would do. We have these blood stem cells. They have this marker on the surface called CD34, so we're able to isolate them. We have our SYNGAP expressing lentiviral vectors, and we have a number of them that we're going to be trying. Some of them are wild type like, some of them have different modifications. The challenge of this approach for a neurodevelopmental disorder like SYNGAP is that SYNGAP is normally found inside of the cell. So we have to sort of manipulate the protein to get it to be transferred from the microglia to the neurons. So there are numerous modifications that we can make. And so we have a number of these SYNGAP expressing lentiviral vectors that we're going to test first for functionality, make sure that they function correctly, and then also for their expression. And so these SYNGAP lentiviral vectors will gene modify these blood stem cells, and then we will have these SYNGAP expressing immune cells, which will hopefully be the therapeutic approach to transfer SYNGAP to the neurons. Once we evaluate these SYNGAP expressing lentiviral vectors in our immune system cells, once we find out which ones function the best, express the most, and all of those details, then we're going to evaluate whether they can correct some of these phenotypes in a SYNGAP mouse model. The way we've done our previous experiments with other diseases is we've first made a humanized immunodeficient mouse model that's disease specific. So there are SYNGAP deficient mice available at JAX, and they're currently being cryo recovered. So they don't have these mice readily available. They're frozen down as embryos. And on their website, it takes about 12 to 14 weeks for them to cryo recover them. We just got the funding for this grant from SRF March 1st. The grant started, so that we ordered the mice right away on day one because we knew how long they would take to cryo recover. So 3 months later, hopefully we will get them. So in June at some point, hopefully. And then what we want to do is cross them with an immunodeficient mouse strain. So this IL-2RG deficient mouse, this IL-2 gamma receptor is used on T cells and B cells to activate them so they can attack foreign material, pathogens. But if you don't have this IL-2 gamma receptor, you're actually immunodeficient and you don't have a functional immune system. And so if we are able to use a mouse model that has this immunodeficiency, cross it with the SYNGAP deficient mice, we can then transplant these mice with human hematopoietic stem cells or human CD34 positive cells. The mice don't have an immune system, so they can't reject these quote unquote foreign cells, which are human. Technically, a immune competent mouse that has a normal IL-2 receptor gamma gene, it has a normal immune system. If we put human cells into that mouse, they will reject them, they will see them as foreign, and we won't get good engraftment, and we won't be able to evaluate our therapy. And for the FDA, for other funding organizations like CIRM, they like us to use as close to the therapeutic candidate that we would use in our preclinical experiments. And so being able to use human cells that have our therapy, our gene therapy in them, is a lot more advantageous than doing everything in mouse cells, gene modifying the mouse cells and then putting them into the mice, because eventually we're going to go into humans and human cells. So if we can skip that step by making this immunodeficient mouse model, then we can use human cells in these mice. And we've We've done this before for our Tay-Sachs work and then our Angelman work, which is published, and it works great. The human cells engraft in. And so once we get these mice from Jack's, we'll cross them with these IL-2RG deficient mice, and then we'll genotype them, make sure that they have this immunodeficiency, and then we'll be able to start evaluating our gene-modified human cells in these mice. So we would take cord blood, CD34 positive cells. We can isolate them for CD34 positive, so we get over a 90% pure population. We're then going to evaluate the therapy in adult SYNGAP deficient mice and then in newborns. We know that the newborn mice that are deficient, they die within a week and a half, but we're able to transplant these newborn homozygous pups at day 2. So we can transplant them with our gene modified cells, and then we can see if we get an extension of lifespan in the newborns. The adults are different. The heterozygous adults will live longer. They do have some behavioral phenotypes, such as a motor, locomotion, and on this rotarod assay where they don't have good enough claw grasping. And so we'll condition the mice, transplant them with the gene modified cells, and then we'll give them 6 to 8 weeks for the cells to engraft. And then we'll start doing some behavioral assays on them, including the rotarod to look at grasping, and then also a number of indications in the open field, including center time. That's more of, are they scared of the environment that they're in? We'll look at locomotion, like how far are they moving around? We'll look at vertical rearings. Can they get up on their hind limbs? And then we'll look at just total locomotion as well. And we'll be evaluating wild type mice, the SYNGAP hets, or the homozygous mice as newborns, pretty much just as an extension of lifespan. If we're able to extend the lives past a week and a half, that's great. And then eventually we could do the behavioral assays. But if we're able to get the newborns to live past a week, then that's significant by itself. We have an empty vector, does not have the SYNGAP gene at all. It's just a control to show that the transduction with the lentiviral vector with an empty lentiviral vector doesn't do anything. And then we'll have our SYNGAP vectors that we're going to use. So currently we're at the beginning of this therapeutic timeline. We are currently developing the vectors, testing the vectors, and then generating the mice. So we've made all of the SYNGAP expressing lentiviral vectors that we want to evaluate, and we're currently titering the last one. Titering is just how much vector is there per volume, so that we know how much vector we need to add to gene modify our cells. We currently have a SYNGAP B cell line that has a point mutation in exon 17 that causes a stop codon instead of an arginine. And so there's no there's not complete expression of the protein, it's truncated. And so we're initially going to evaluate the function and expression of our vectors in this SYNGAP B cell line. And then we'll look at expression of these vectors, and then we'll also look at functionality in these RAS and GTPase assays, just to see if the modifications that we made to some of our vectors, if SYNGAP is still functional in its normal activity. And so it is at the beginning of this therapeutic timeline, but once we have the mice generated and all of these experiments, the functionality and expression and all that will be done prior to when these mice will be made. It just takes a long time because they had them cryo recovered. And so it's 3 months just to get those mice out of the freezer. And then we'll start on the efficacy experiments. And then if we see correction of some of these clinical phenotypes, then we'll move on to the safety experiments. One of the big potential risks with what we do is that we're overexpressing these proteins in cells. And so will it cause some detrimental effects on engraftment of these hematopoietic stem cells, on generation of the T cells, B cells, microglia that are made from the stem cells? And then will those cells function? Because it's one thing to express these therapeutic proteins, but if we're causing detriment to the immune system, then there will be other problems. The way that this would work for a clinical trial, it would be like a normal bone marrow transplant. So there are mobilization drugs. Most of our hematopoietic stem cells live in the bone marrow. As I mentioned before, there's a small percentage that's circulated in the blood, less than 1%. And so we wouldn't be able to get enough stem cells out. So what the patients get is a mobilization regimen, which actually pushes the stem cells out of the bone marrow into the blood. And then the patients just get a blood draw, just like if you're going to donate blood. They sit on a machine, and then cells are taken out of the blood into a leukopak. And then what we do, the arrow pointing down, is that we will then isolate those stem cells to greater than 90% purity. And then we genetically modify them, the stem cells, with our SYNGAP vector ex vivo, so outside of the body. And then a patient gets a conditioning regimen so that they can make room in the bone marrow for the stem cells that we then want to transplant. And then the patient just gets an intravenous injection of the gene modified SYNGAP expressing stem cells. These hematopoietic stem cells know where to home. We can put them in the blood, but they know to go back to the bone marrow. And so we don't actually have to do anything to the bone marrow itself, as far as like sticking a needle in the bone marrow, taking bone marrow out that way, or putting the cells back in. Everything can be done IV. And so the reason why we wanted to test this approach for other neurodevelopmental disorders is because of what we saw with our work with Angelman syndrome. This work was published in collaboration with Jill Silverman's lab last year, where we showed that we could both prevent and rescue clinical phenotypes in an immunodeficient humanized mouse model of Angelman syndrome in both adults and newborns. The adults typically don't show clinical phenotypes till about 6 to 8 weeks. And so what we did was we transplanted these mice both as newborns and as adults once they started showing phenotypes. And then we looked at open field locomotion, beam walking, gait correction, which are all sort of motor and behavioral assays. But then we also looked at novel object recognition. It was more like a memory and learning. And then we also looked at a decrease in an EEG delta wave spike. So in Angelman patients, they have a large jump in their delta wave when you do an EEG. And what we saw, we didn't see a correction all the way down to wild type, but the spike was cut in half. And that was significant to actually show that we could take this delta wave down. And so after seeing all of this, for Angelman syndrome, we are now looking at other neurodevelopmental disorders to see if this approach could work. Similar to SYNGAP, UBE3A, which is the protein indicated in Angelman syndrome. It's an intracellular protein. It's a ubiquitin ligase, so it doesn't normally get secreted out of the cell and taken up by other cells, unlike Hex A and Hex B for Tay-Sachs or some of the other proteins for the lysosomal storage diseases or leukodystrophies. So we had to make some modifications to UBE3A to allow it to get out of our gene modified cells. And some of those similar modifications we're testing in SYNGAP. But this is the reason this publication, if anyone wants it, I can email it out. It's freely available online. Anyone can access it and look at it. But that's the main reason for our endeavor into SYNGAP, is because of how well it worked for Angelman syndrome. We wanted to look at it for other neurodevelopmental disorders. So I'll just go through this quick. It's just showing a little bit of the data from the Angelman syndrome work. You know, same setup as far as the mouse model. We made a immunodeficient humanized mouse model, took cord blood, gene modified with our UBE3A vectors, busulfan for adults or radiation for the newborn mice. And then 8 weeks later, post-transplant, we did behavioral assays. And then I'll show some data for that. This is an open field assay. The blue line, so the mice go in this box and then they just walk around and they explore, and it has beam breaks, so you can tell how far they've traveled, how many vertical rearings on their hind limbs, how much center time. It actually calculates how much time they're in the center or how much time they're on the perimeter. And then we can look at total activity. So the blue line is our gene modified cell transplanted mice. This top line is wild type. And then these bottom two lines are the UBE3A deficient mice. And we saw some correction of this total activity. Oh yeah, the video shows the mouse going around. So this is what we'll do for the SYNGAP mice as well. This is a beam walking assay. The increased number of rod number actually decreases the diameter, so it's harder for the affected mice to cross rod 3 because it's a thinner rod, so they take more time. The blue again is our UBE3A vector transplanted mice, and we can see that these mice on rod 3 acted similar to our wild type mice, where these two higher bars are our UBE3A deficient mice. This is the novel object recognition. There's a familiarization phase where there's a similar object, and then when you throw this novel object in there, the normal mice will actually go and sniff and hang out around that novel object a lot more than the disease affected mice. And we can see that in this blue bar here with our UBE3A vector transduced cell transplanted mice. They actually spend more time around that novel object, similar to the wild type, compared to the UBE3A deficient mice. And then this shows the delta wave spike. So in humans, an AS child has this red spike in the delta wave, where a non-Angelman child has this normal delta spike. And then in our mouse model, this lower line is our wild type mice. The blue line is our vector transduced cell transplanted mice. So you can see that it was almost cut in half from the spike of our UBE3A deficient mice. So seeing this was pretty significant that we were able to take that delta wave down. So where we are for this project, we're currently doing the IND enabling studies. The FDA, for an adult trial for Angelman's, they wanted us to do one more toxicity and expression study. So we're currently doing that. But then for a pediatric trial, they wanted us to do 3 more efficacy experiments: sleep disruption, seizure threshold, and 1 more. And so in order to show direct patient benefit for a pediatric trial, we have to complete these 3 additional efficacy studies. And then for the adult trial, which might start sooner than the pediatric trial, they might just be separate trials, just depending on how the experiments go. We're just doing 1 additional toxicity and expression study, which is going to be similar to what we would have to do for SYNGAP if these vectors work in the SYNGAP mouse model. So I'd like to thank the people in my lab, Julie and Camila, and then everyone in the vivarium, which is where the mice are kept, for all of their work. And then of course, everyone in the Silverman lab that worked with us on the Angelman syndrome study. And then I'd also like to thank the SYNGAP Research Fund for funding our current research for the SYNGAP therapeutic approach. And thank you. I'll take any questions.
Awesome, Joe. Thank you very much. I've got a couple of things off the top I want to say for the families who are watching, because great presentation. It feels a little sci-fi to some people, and I just want to emphasize to people that you have made good progress in Angelman, and people who follow our work at SRF know that I follow Angelman very closely. And on the one hand, this work, if successful, would require our kids to go through a bone transplant, a bone marrow IV infusion, which sounds a little scary to people. But could you speak to the relative theoretical efficacy of this versus, say, an ASO or an AAV in terms of percent of brain that would be affected? And I realize that this is a theoretical conversation, but it is an interesting feature of this approach. Do you know where I'm going?
Yeah, so the microglia, these accessory cells, are found throughout the brain. And so by transplanting the blood stem cells that have this gene modification, we potentially could reach every part of the brain that these microglia are in. They're also going to be, I guess you can call them protein factories, where they're just going to be expressing this protein and secreting it out of their cells, whether into space or in direct contact. We know that in neuronal damage, there is direct contact where these accessory cells like astrocytes and glial cells are in direct contact with these neurons. So they could actually just be sending it across the cell membranes and into the neurons. But the fact that these microglia and accessory cells are found throughout the brain, there's, I think, a greater potential to send these proteins to more neurons compared to an AAV or an ASO, where it might be injected in multiple parts of the brain, but is it going to get to a majority of the affected neurons? I don't know that, but just knowing that the microglia are found throughout, I think this approach has the potential of affecting more of the neurons. And then plus, like I said before, we're not directly gene modifying the neurons themselves.
Yeah. So then there's a few science questions pouring in, and before I defer to them, I want to make another point. So we are funding the early work on this, and as you pointed out, so frustrating, we have to uncryo these mice, and it takes so long. But the sooner we start, the sooner we get somewhere, right? And then if this works, there's a lot more work to be done, and the $130,000 for this initial experiment really is just the tip of the iceberg. So one of the first conversations you and I had was, oh, who's going to keep paying for this? And hopefully, there's enough good evidence that we want to keep supporting it at whatever level we can. But can you talk a little bit to potential other funders as this work gets its sea legs, like California Stem Cell, what are they called?
Yeah, so the California Institute for Regenerative Medicine, they have a huge neurodevelopmental pipeline that they want to develop. And if we're able to show efficacy in the mice, we should be able to get funding from whether it's NIH, whether it's CERM. If we're able to show it in the mice, then I don't really see a problem of getting funding, especially with something that would work this well.
Yeah, exactly. So I think the point I'm trying to make for for the families is this will take a lot more time and a lot more money, but
Yes.
hopefully our money will be catalytic and could support the creation of more money for this work, point one. And point two, even though it is far off, and we have now 2 companies publicly working on ASOs for SYNGAP1. Just yesterday, Praxis announced they're working on this. I think the potential to have a one and done treatment that would, as you just said, create a protein factory in the body is just too tantalizing not to go after, in my opinion.
Right. Yes.
And that's the point I want the families to land on. There's a lot of questions here. There's a lot of what about this and what about that? But the punchline is, if we can pull this off, there's thousands of Syngapians out there. We got to help them. So with that, I put Hans up on the panel. Hans, do you want to ask your question? And then, I want to honor one more question, then give it to Hans to handle the science stuff. Pat Vreeland said, if all goes well, how many years are we away from dosing a human in a trial? Big if, but
it will probably take 8 to 12 months to evaluate this in the mice. We then have to do, if we get funding right away for the toxicity, then that would take another year or so. I don't know. It's really hard to say. If everything goes well, I would say 3-4 years, depending on funding. But everything has to go perfectly. The big thing right now is the mice, getting these mice up. We still have to cross them with the IL-2 mice. That takes generations of mice to make, so that's months. Because once you cross a wild type IL-2 with a homozygous IL-2 deletion, you just get heteros, and then you have to cross those heteros to get your homozygous mice, and then you have to grow up those homozygous mice to then get enough to do the experiments with. And so getting this mouse model up and running is the first thing. We may actually do some experiments that I didn't put in the initial grant where we use the mouse cells and go into the mice while the humanized mice are being made. We'll have these immune competent SYNGAP hetero mice that we could transplant. So we could get some initial indications. Those experiments are quick and easy to do while these immunodeficient mice are being made.
Got it.
So yeah, but it's just hard to say. What's the FDA going to come back? Are our open field and rotarod assays enough to do an adult trial? For our Tay-Sachs, our Sandoff, that was enough. We just had to do a toxicity and expression study, which we're doing now for Tay-Sachs, and then we're able to put in our IND for an adult trial for Tay-Sachs. So it might be enough, and that's a big if as well. We thought we did enough for Angelman's with all of that efficacy to be able to do a pediatric trial, but they want us to do 3 more. It was enough for an adult trial, but not for pediatric trials. So it's hard to say.
No, it's a hard question, and I am not asking it to push you to give us an answer we're going to be happy about. But just to highlight for the audience that the science stuff takes time and it's unclear. But again, the sooner we start, the sooner we get somewhere. Okay, Hans, you want to do the science questions?
Well, Joe, it's good to speak again. And
You too.
Thank you. To some degree, I've asked these questions before, but we just have to be so careful. Can we talk further about the malignancy risks, in the sense that the mice are treated with busulfan to condition them for space in their bone marrow for the engraftment? Is that right?
Correct.
And are there, is it similar in the human then in terms of conditioning regimen? And are there malignancy risks secondary to the conditioning?
are actually moving into a new conditioning regimen. It's actually a CD45 antibody method that is safer, less toxic, and it actually clears more of the bone marrow out. So they wouldn't get busulfan. There's already trials that have used it. We're actually adding it into our HIV trial for our next trial. And so there are safer ways. As far as the conditioning regimen causing a malignancy, there's always that potential, especially with chemo conditioning regimens, but with this 45 antibody, it's different and it acts in a different way.
Yeah.
So I can't really speak as far as potential malignancy for that because it's more new. The FDA is always concerned about integration status of a lentiviral vector. Where is it integrating this and that? The fortunate thing is that HIV, which the vector is based off of, there's been no report of it causing a primary malignancy. Obviously, it causes other ones because you decrease the immune system response and you get all these other cancers. But as far as where lentiviral vectors integrate compared to retroviral vectors, they have a different integration pattern. We do those experiments. That's part of the toxicity in the mice. We also do these immortalization assays in vitro where we look at transducing the cells and then passaging them for a long period of time and look for outgrowth of these cells just based off integration pattern. We also do integration analysis on where these vectors integrate, and we take a sample of different cells that have been transduced, gene modified, and we do an integration analysis and look for any hotspots. We've done that for others and we don't see it. But yeah, it's always something that we're looking for and is in the back of our heads because the vector integrates and it's permanently in there.
And that harkens back to the SCID trials, right? I mean, the kids who were getting gene therapy for severe combined immune deficiency, there was some malignancy signal there. But the more recent, bluebird bio, AML, MDS, it sounds like the treatment itself has sort of been exonerated.
Yeah, and so for the SCID, those were using retroviral vectors. So they integrated near a gene called LMO2, which is an oncogene. And with these retrovectors, some of their LTRs are active. And so they integrate near a gene. The LTR is the normal mouse retrovirus promoter. It can upregulate nearby genes. So the LMO2 gene was upregulated, and that's what causes the cancer. But now people have moved into lentivectors. With the bluebird, yeah, that was just hard to say whether it was the promoter they were using, the gene they were expressing, whether some of those kids were predisposed to any of that. What kind of conditioning regimen? I haven't heard anything yet as to what actually caused the MDS. I know the kids were treated for the MDS and they recovered, but it was a small percentage. I think they had done at least 60 or 70 kids or 80 kids, and it was in the low single digits. I don't know. Are those numbers correct? Is that what you've heard? Like 3 or 4 of the kids had MDS?
I thought it was in fact lower, even just 1 or 2. And then there was an AML that was written up in the New England Journal in January.
Okay, so yeah, one thing that would be good. So what we can do is we can actually sequence a person's genome and actually look for any of these predispositions. And I was talking with our bone marrow transplant physician because we use the similar promoter that they suspected was causing this in our Tay-Sachs vector and our Angelman vector because it's such a good promoter on expressing proteins. And they wanted me to change it out. And so we've been changing out this promoter for other high expressors just because there's that potential. And then I was talking to him and was like, well, maybe we should sequence some of these patients prior to enrolling them, see if they have any predisposition to any of these diseases. Sometimes that's done with these lymphoma patients that we have in our HIV trial. We actually sequence them and see if their lymphoma is caused by a specific mutation that they had. And so we're able to look at predisposition of some of these diseases based on sequence. And so if it is something about these conditioning regimens or something like that, there's potentially things we can do pre-enrollment into these trials to see if there is any, potential for malignancies.
Well, I have to admit, to hear that there's, an improved conditioning regimen, an anti-CD45 is so much more appealing than some, caustic chemotherapy agent.
Yeah, and the fact that it clears more out, so we're able to get a higher level of engraftment. For some of these monogenic diseases, we may not need the large level of engraftment of gene-modified cells as we might for our HIV stuff. So sickle cell, I think they only need around 15% gene modification to actually get correction of phenotypes from sickle cell, because then you get enough of those red blood cells that are corrected, so you're able to function normally.
Yeah, it's probably lower hanging fruit in terms of correcting that phenotype. But I hear you. Oh, it's fantastic.
Can we get to some of the science questions? There was one in the chat from Shin Chen. To what extent can microglia graft in the CNS? Is that?
We are kind of looking at that. We're doing immunohistochemistry on the brains of our transplanted and engrafted mice, and that's actually part of our SYNGAP proposal. We have that in there, so we can look and see how many of the human microglia engraft into the brain. We're currently doing that with our Angelman. With a conditioning regimen and a strong one, I feel like we can get a large number of them, but we will be looking at that. We'll be doing co-localization of human microglia and mouse SYNGAP in our studies. So we'll be able to look at those and quantify them.
Great, thanks. And then Ray Holden asked a question. I'll let Hans parse. There's a number of other questions from JJ in here, but I'm gonna just throw Ray's at you. I'm not quite sure what he's saying, to be honest, but I don't want to talk anyone out of a job or research, but would parent to child stem cell donors create corrected glial cells, or are the modified glial cells imperative to the success of this method? And isn't the answer that you need to modify them to have them make the SYNGAP? Or am I making that up?
Can you repeat the part about parent to child? I can't find that question in here.
You can't see it. If you scroll down the open, it's about halfway down. If you scroll down on the Q&A.
It's in the Q&A, and I think it's, naturally a dad.
Oh, in the Q&A. Okay.
Yeah, a dad is interested in giving his cells, and not putting his child through the effort. But then you've got a degree of mismatch.
Oh, yeah.
And
I don't think that would work. That would be an allogeneic, and there's actually more toxicity in that, graft versus host disease, all of that, compared to just using autologous cells on a patient's own cells. So just as a sort of maybe a little bit of relief, Bluebird's done this in children with their cerebral adrenal leukodystrophy, and Don Cohn at UCLA has done this with the ADA SCID kids. So this is done all the time in kids and juveniles, and the protocols are there. So I would be confident and I would feel comfortable if I had a child, because it's been done so many times and the protocols have been there.
And to the point, it's going to be a few years until we're doing this to humans, assuming all goes well. And in those years, we would expect the protocols to get even more refined, much like the the clearing regimen you were talking about becoming a better choice, right? So
Yeah.
the only thing we can be sure of is that science continues to happen at lightning speed. So the fact that we're a few years out in that sense is good news, because things should get more refined. Hans, you want to manage the rest of these?
Well, so one of them is how are you going to control where the SYNGAP1 lentivirus integrates? And it's not something under your control, but the lentivirus integration sort of profile is seemingly benign.
Correct. Yeah. And we've done these integration analyses on our patient samples for our HIV trial. And there's actually over 13 to 15,000 integration hits. They can be in introns, exons, close to CPG regions. They're just all over the place. But where they like to integrate compared to where the retros like to integrate is completely different. And plus the vectors are inactive. The ends of the vectors that actually do the integration, they have no promoter activity. There's a poly A tail in the 3' LTR, so it stops any transcription from going through the vector. So the only thing that's in there that's active is our promoter that's driving the expression of SYNGAP. So these self-inactivating lentivectors have been designed to not cause much damage once they integrate in. But yeah, we cannot control where our specific vector integrates.
And then there's a question of whether there's toxicity observed in the preclinical work, whether it's for Angelman's or another disease.
We have not seen anything yet in our Angelman mice. We just got our adult toxicity data back, and there was nothing that we saw that was toxic. As far as this one, we just don't know yet. We'll have to do those experiments. We'll get an initial idea when we actually transplant the SYNGAP hetero mice. If something happens, whether it's weight loss or some other thing that we observe, then that could show some potential toxicity, because those could technically be called our clinical trial patients because they have the SYNGAP deficiency. So we'll be able to get some idea once we start transplanting the mice.
And so have you done a second species as a part of the safety studies for Angelman treatment?
No, I think because we use the human cells and we use these immunodeficient mouse models that we haven't had to go to non-human primates. We weren't asked for HIV, we weren't asked for our Tay-Sachs, and we weren't asked for our Angelmans in our pre-IND meetings with the FDA. So I think using these humanized mice, where we're actually using the human cells and not just mouse to mouse, that actually helps.
That's fantastic. Thank you for your patience. Mike?
Yeah, this is great. I just wanted this one question up that I just want to cover. I think we talked about it, but how are you going to get SYNGAP1 protein into neuronal cells?
So if we go back to the,
it sort of bears repeating if people are unclear, right?
Yeah. So if we go back to the second slide, the way the hypothesis works is that the microglia that are transplanted and then engrafted into the CNS will act as protein factories. So in the upper left corner, that's our gene modified microglia. And then in the bottom right, that's our affected neuron. And just based on modifications that we're making to the protein, we should be able to transfer it to the neurons, whether in the extracellular space where the microglia express it, and then the neurons take it up, or with a direct contact. So with neurodegeneration or neuron injury, sometimes there's direct contact with these accessory cells like the astrocytes and the glial cells. And so it could just get transferred in between the cell with close proximity or cell membrane contact. We don't know the exact mechanism, and I don't know if we'd be able to identify that just with some immunohistochemistry at looking at expression. One cell takes it up, one cell expresses it. We see that co-localization, but how it's actually getting there, we just don't know that exactly. But knowing how the cells work and interact and what we're doing to modify the proteins, there's a couple hypotheses. One might work, other one might be right, but they both might be right. But yeah.
Got it. And just for the people on the call who don't think about neuroscience all day long, the people like me who've been trying to figure it out for the past 3 years and are still struggling. Broadly speaking, there's at least two kinds of, there's neurons and there's glial cells in the brain, right? And so what you're talking about is affecting the bone marrow to create the glial cells, which would produce what we need to go from the glial cells into the neurons. And the neurons is where we find SYNGAP. Like at that sort of cartoonish level, is that accurate?
Yes, and then what we are hoping is that once SYNGAP gets into the neuron will know where to put it. So it would be in the synapses. So, normally SYNGAP is made, the RNA is made in the nucleus, just like any normal cell, and then the protein is made, and then it gets transported, whether it's a cell membrane protein, it has cell localization signals in it so that the cell knows where to transport it. So we are hoping that once SYNGAP gets into the neurons, the neuron will then put it where it needs to go. At that point, we have no control because it's in the neuron and the neuron's going to do what it wants to do. But it should put it in the right spot. It should have all the correct signals. We don't modify any of the major like reactive regions, the enzyme active regions, any major structural regions in the proteins. We try and stay away from all those active regions or binding domains, things like that. And we just make these modifications in other regions, maybe in some like other folds that don't seem to be necessary just based off of current research and what we know about the proteins.
Right. So, I mean, so I'm just going to editorialize for a hot minute. I went to the Angelman meeting where you presented this work and you've been driving this, and I talked to a lot of people, and there are some people who are like, well, there's still some things we don't understand. And there are other people who are like, yeah, that's kind of how science works. But we're seeing things in the mice. And if this does work, the thing that struck me and where I really decided we needed to fund this, I talked to one very smart Angelman parent. And I said, it was off the record conversation. I said, what do you think of this? Like, tell me and she said, I would want my child to be patient one because it's one and done. And she said, if this works, and it's a huge if, but she was very enthusiastic. And I'm not saying that you're going to create a silver bullet here, but I am saying that I think the purpose of the SYNGAP Research Fund is not to fully fund the creation of entire therapies. That is just a stupid amount of money. The purpose of the SYNGAP Research Fund is to make sure we leave no stone unturned. And when we find something like this that could be a winner, and we find someone like you who's courageous enough to push the ball up the hill, I think we owe it to our kids to support you until you can get enough data to get the NIH and the California Institute for Regenerative Medicine and others to keep funding the work. So one of the parents asked, how do we support this work? The answer is simple. You help us, you raise money for SRF, and you help us support Joe and other people who have the courage to go after this stuff. So thanks, Joe. Keep it up. I know it's tough to get up here and be like, what about this? But we are grateful, and we look forward to working with you.
Yeah, thank you. And thank you for funding the research, because it could be this, it could be something else, it could be a combination. We just don't know. And when there's nothing currently out there, we're grateful and appreciate the opportunity to, test our theories and our hypotheses on what could happen. It seems like it would work just based on what we know of the hematopoietic system. And, you have a cell that doesn't have a protein that works. So we need to figure out how to get that protein into the cell. Once we get that protein in there, hopefully it does what it needs to do and we can correct these neurons. But yeah.
Okay. Thank you very much, Joe. Good luck.
Thank you.
Thanks, Joe.